Hi students, next topic is conformation of DNA. Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment. Dear students, DNA is deoxyribonucleic acid is a hereditary or genetic material. Studies on DNA of various bacteria, viruses and eukaryotes have given rise to a number of surprising findings. A number of variations still exist with respect to the structure of DNA that serves as genome material in viruses, bacteria and eukaryotic organisms. Three major forms of DNA are double-stranded and connected by interactions between complementary base pairs. They are A form, B form and Z form of DNA. Some other forms of DNA like C form and D form of DNA is also present. So in this session we can discuss about the conformation or various forms of DNA in detail. Most of the DNA is in the classic Watson-Crick model, simply called as bDNA or B form DNA. In certain conditions, different forms of DNAs are found to be appeared like aDNA, zDNA, cDNA, dDNA and eDNA. This deviation in forms are based on their structural diversity. These are the different forms of DNA based on X-ray analysis. bDNA, most common, originally deduced from X-ray diffraction of sodium salt of DNA fibers at 92% relative humidity. aDNA, originally identified by X-ray diffraction of analysis of DNA fibers at 75% relative humidity. zDNA, left-handed double helical structure, winds to the left in a zigzag pattern. cDNA, formed at 66% relative humidity and in the presence of lithium and magnesium ions. dDNA, rare variant with 8 base pairs per helical turn, formed in structure devoid of guanine. eDNA, extended or eccentric DNA. This slide shows the double helical structure of DNA. First we can discuss about bDNA. Described by James D. Watson and Francis Crick, commonly found DNA. DNA molecule consists of two helical polynucleotide chains coiled around common axis. Two helices are wound in such a way so as to produce two interchain spacing or groove. Major groove with 12 angstrom, depth 8.5 angstrom. Minor groove with 6 angstrom, depth 7.5 angstrom. These grooves provide surface with which proteins, chemicals, drug scanning interact. So this is the double helical nature of Watson-Crick form of DNA. Two helical wind along the molecule, two chains run in opposite direction, they are anti-parallel, plane or bases are perpendicular to helix axis, right-handed twisting, uniform diameter, complementary base pairing, base pair per turn is 10.4, rise per base pair is 3.4 angstrom. The next one is aDNA. It is one of the possible double helical structure which DNA can adopt along with other two biologically active helix structure like B and Z DNA. Right-handed, short and fat compared to bDNA occur only in dehydrated sample of DNA such those used in crystallographic experiments. aDNA was originally identified by X-ray diffraction analysis of DNA fibers at 75% relative humidity. The grooves are not as deep as in bDNA. The bases are more tilted. The base pairs per turn is 11. Rise per base pair is 2.3 angstrom. This is the three-dimensional picture of aDNA. Next is zDNA. One of the many possible DNA double helix structure. Left-handed double helix structure wind to left in zigzag manner. So they are termed as zDNA. Discovered by Rich, Northam and Wang in 1984. It has anti-parallel strands as bDNA. It is long and thin as compared to bDNA. The remarkable characteristic is adjacent sugar have alternating orientation. In zDNA, purine, conformation based sugar are near or and on same side. Pyrimidine anti, that means bases and sugar are distant on opposite sides. Only one deep helical grooves. There are 12 base pairs per turn with axial rise 3.8 angstrom and angle of twist is 60 degree. See, this is the various representation of Z form of DNA. Next is cDNA. It is formed at 66% relative humidity in presence of lithium or magnesium ion, right-handed, with axial rise of 3.32 angstrom per base pair. Helical pitch is 3.32 angstrom cross 9.33 angstrom. Base pair rotation 38.58 degree, has a diameter of 19 angstrom, smaller than that of A and B DNA. The tilt of base is 7.8 degree. This shows cDNA. Next is ddNA, extremely rare variant with only 8 base pairs per helical turn. Axial rise of 3.03 angstrom per base pair. Actually, two different forms of ddNA. ddNA A and B. A take part in DAB transition, B associated with the DB change of conformation. Two DNA structures have same helical parameters. This is the ddNA structure devoid of guanine. Next is eDNA. Cytosine methylation or bromination of DNA sequence is to induce a novel extended and eccentric double helix, which we call eDNA. It has a long helical axis rise and base perpendicular to the helical axis, deep major and shallow minor groove. eDNA allowed to crystallize for a period time longer. The methylated sequence forms standard aDNA. eDNA is the intermediate in the transition to aDNA. eDNA is the intermediate in the crystallographic pathway from B to aDNA. See the diagram of eDNA. This table shows various characteristic features of various forms of DNA. Shape, helix length, helix diameter, then condition, rise per base pair per turn, helix pitch, rotation, base pair tilt, glycosidic bond, major groove, minor groove, etc. Dear students, I hope you could understand various forms of DNA that we discussed. Thank you for watching. See you in next class with another topic. Don't forget to leave a like and please subscribe my channel.